But that hasn't prevented research from continuing. In 2024, a huge collection of nematode researchers released the paper, "The Time is Ripe to Reverse Engineer an Entire Nervous System." And they specifically talk about C. elegans, the nervous system of the nematode. And the reason that they are so compelled by it is that we do definitely have a huge collection of data that we can actually derive from, which I will go over soon. But we seem to have it all. We seem to have the compute. We seem to have a bunch of data sets. But the question really is, before we get into all that, why? Why would you simulate such simple, small animals? And the answer is that what we need is something that proves that, that we know what we're talking about when we do neuroscience simulation. If we claim that our models are representations of what's really going on, then let's prove it. Let's demonstrate that we can actually take a nervous system, this entire one that we have here. We have the knowledge of the entire body, all the muscles, all the reproductive organs, everything, and put it all together in a simulation and demonstrate that we can actually reproduce behavior from the brain. If we can't do this for a 302 neuron microscopic animal that's just a worm, then how can we ever dream to do it for 100,000 neuron fly or for a multi-million neuron mouse or eventually for a 10 billion neuron human? Just understanding the difference in these scales, we have a long ways to go if we can't even simulate 302 neurons and show how they manifest in behavior. So there is a new paper, another 2024 paper that came out to try to at least do it with the neurons of the motor system. Sort of, I guess, a successor of the open worm initiatives. And I'll show you, first of all, before we visit that, I'll show you kind of my version of what the nervous system simulation might look like. Now, these are not really connected neurons, and the actual biophysical properties of every neuron are not exactly real, but you can see what it might look like. So it would be a bunch of neural activations. Theoretically, they should be activating each other. They should be getting activated by the environment and stimuli, and then triggering all the behaviors that we see worms do. And in theory, you should be able to track the activity of every bit of this nervous system and correlate it to the behavior of the nematode. So, starting from these guys, we can take a look at their particular version of this. You can see a sort of crazily squirming worm. So they've created a sort of muscle model, and they've ripped out the part of the nervous system that's responsible only for the muscle activity. And they've tried to see if they can use it to drive the worm towards food and to create the sort of natural squirming behavior we should see. Now, we have actually a lot of recording data of real live worms moving around. And so hopefully we should be able to try and match a simulation like this to real behavioral data. Okay, so that's all well and good, but really, what's left then? What are we even talking about? Well, the answer is that we don't really have all the data, nor do we know that just replicating some muscle dynamics gets us anywhere close to the real nervous system. So we can start with Wormbase, which is one of the oldest projects. If we go back to the history, Wormbase was spawned in 2000 in Caltech. And from here, you can actually grab the 3D model of the worm. You can see it was also the original progenitor of this browser atlas thing that I used in 2011. And so Wormbase is really quite literally the base of a lot of how the worm stuff came to be. But there were also other databases. We saw the genetic one before, but there's Worm Atlas, which has a lot of the great data. And in fact, this diagram here, where you can see every neuron in this graph and connect it to the body, well, that is from Worm Atlas. But it has a lot of other stuff, single neuron data, things about different cells and cell lineages, and just about any sort of, it's like the encyclopedia or atlas of worms. There's also Worm Book, which is an additional sort of collection of pieces of information about the nematode, biochemistry, signal transduction, genomics. What we really see when we look at the real data is that this thing goes really, really deep. 302 neurons and a couple thousand cells can get really, really complicated. Just as an example, here is a list of all the different neurotransmitter receptors, and this was a list only in that was last revised in 2013. And so you can see that, they have a lot of different ways of transmitting chemical information. They use different neurotransmitters, many of which we ourselves use: glutamate and GABA, acetylcholine, dopamine, serotonin. You'll find that there's a lot of overlap. In fact, they even have, I don't know if I'd be able to find it right now, but they even have the same receptor for insulin that is targeted by things like Ozempic.